The basic idea that Dan had is that he wanted to do metabolomics. So we have the ability to measure metabolites in the tissues of mice.
Mhm.
And he did this in mice with the different mutations, but also under the different conditions of calorie abundance.
And when you say you did metabolomics, are you looking in a particular tissue here? So my understanding is you're looking at the hypothalamus. So you're kind of looking in the region
Right.
of the brain where this core conductor, the central clock lives. And when you say metabolomics, you basically mean you can do more or less an unbiased test to see how all, or at least many of the metabolites in this tissue in the hypothalamus are changing. So you can take a pretty wide and pretty unbiased look, everything that is changing in this key regulator of circadian rhythms in the hypothalamus.
Exactly. And so I don't remember the exact order, but you can do it in the three different lines of mice that we have. The FASP mouse, the wild type mouse, and the one that has the amino acid that's negatively charged. And there were dramatic differences, particularly in fatty acids, that could be measured when he isolated hypothalami from those mice, ground it up, and sent it off to a metabolomics lab where they use sophisticated techniques to look for, certainly not all, but many, many metabolites that we have in our normal cells. And he found that the most dramatic differences between the three different mutant lines. Let's talk about the FASP mutant line, which is the one that has the human mutation.